Hey there, curious minds. You know that feeling when you've finally fought off a really nasty virus? You're technically better,
Hmm.
but something just feels off,
Yeah, definitely.
especially maybe with your breathing. Well, for literally millions,
Hmm.
the sort of lingering aftermath. It's often called PTSD, post-acute sequelae of SARS-CoV-2.
Right, PS, yeah.
It's become this huge public health challenge. But the really baffling part, we haven't really known why these long-term issues, particularly in the lungs, just stick around. It's been a bit of a mystery.
It really has. And while we've seen growing evidence, pointing towards maybe abnormal immune responses or perhaps the organs just not recovering properly,
Uh,
the exact links, the step-by-step mechanisms connecting these things in PSC, they've been frustratingly unclear. It's been a major gap, honestly, in how we manage it clinically.
Absolutely. It's like, okay, we see the problem, we know it's stubborn, but we don't get the root cause.
Hmm.
So our mission today, we're going to take a deep dive into some really, really groundbreaking new research. It's a paper just published in Nature, and it's starting to pull back the curtain on this whole complex puzzle. We're going to unpack exactly what's going wrong in the lungs.
At the cellular level.
Exactly how it's happening cell by cell, and maybe the most exciting part, what this could mean for potential new treatments. Treatments that could actually rescue lung function after the infection is long gone.
Which is huge.
Yeah, get ready for some serious insights, I think. Okay, so let's start with the big picture. When we talk about PSC, or what lots of people just call long COVID,
Right.
what are the specific lung challenges we're actually seeing? And why has pinning down the cause been so, well, difficult?
Well, the long-term effects, the physiological consequences of these respiratory viruses, particularly post-COVID, they're pretty expensive. Patients often report things like persistent shortness of breath, maybe a chronic cough that just won't quit, and
Yeah.
really noticeable reduced exercise tolerance. Like they just can't do what they used to. And in some really severe cases, unfortunately, we see progression to serious lung fibrosis.
Fibrosis.
Yeah.
Can you remind us exactly what that is?
Sure. Fibrosis is basically scarring. It's the formation of excessive scar tissue in the lungs. So imagine your lungs, which should be soft and spongy, becoming stiff, almost like a rigid sponge.
Ah,
And that means they can't expand properly, they can't exchange oxygen efficiently. So the big challenge has been that the cellular and molecular etiologies, fancy word for the origins or the causes.
well, why?
Exactly. The why of these lingering issues have been really poorly defined. It's like, we could see the smoke, but we didn't understand the exact fuel or the ignition source of the fire.
We had the what, but not the how or why underneath it all.
Precisely. The detailed cellular roadmap was just missing.
So this big gap in understanding, how did this new study actually try to fill it? What was their approach to start drawing that missing map?
They used a really smart, sort of comprehensive strategy. First off, they gathered insights from 3 different groups, cohorts, we call them, of actual patients who had respiratory PASC.
Okay, real world data.
Exactly. Invaluable stuff. It showed them how the problem actually looks in people. But then to really get down to the nitty-gritty, the underlying mechanisms, those intricate cellular conversations, they also developed a really robust mouse model of post-viral lung disease.
Okay. The controlled environment.
Precisely. And that combination was powerful. It let them go from just observing the problem in humans to really identifying the cellular and molecular roots in the lab. They could, tweak things, isolate factors, understand cause and effect.
It lets you ask why in a way you can't always with just observation.
Exactly. That's how you get from what to why.
Okay, let's really unpack this then.
Yeah.
The paper jumps right in, talking about identifying an aberrant immune-epithelial progenitor niche unique to fibroproliferation and respiratory PSC. Wow, okay, that's a lot to unpack.
Huh. It is a mouthful, isn't it?
Yeah. Can you break that down for us, maybe piece by piece? Like, what's a niche in this context?
Absolutely. Let's start with niche. Think of it like a specific little microenvironment in your body, almost like a tiny neighborhood or a mini ecosystem.
Okay.
And in this neighborhood, certain types of cells live and interact. And these interactions are fundamental. They basically dictate whether cells grow or heal or differentiate.
So it's about the local environment and the neighbors.
Exactly. And here, we're talking an immune-epithelial niche. So that means the interactions are between immune cells, our body's defenders,
Right, the infection fighters.
and epithelial cells. Those are the cells that form the vital lining of our lungs, like the inside surface of the air sacs, and specifically, they're progenitors.
Progenitors. Like
Sure.
stem cells?
Sort of, yeah. Think of them as precursor cells. They're the lung's natural repair crew. They're meant to regenerate, patch up damaged lung tissue after an injury.
So the lung's own repair crew, living in their little neighborhood where they should be getting the right instructions to rebuild things properly.
You got it. But here's the absolute key word, aberrant.
Aberrant, meaning abnormal, wrong.
Exactly. Abnormal. Dysfunctional. The interactions happening in this niche are not leading to healthy repair. Instead, this specific aberrant niche is uniquely linked to fibroproliferation.
Which you said is the scarring.
Precisely. The excessive growth of fibrous, scar-like tissue. So going back to your analogy, imagine the lung's repair crew, those epithelial progenitors. They're not getting the right blueprints.
Okay.
The signals from their immune cell neighbors are wrong. So instead of smoothly patching things up, rebuilding healthy, flexible lung tissue, they're just kind of haphazardly laying down sticky, stiff concrete.
Oh, wow. So it's not just damage, it's faulty repair leading to more problems, a dysfunctional neighborhood.
That's a perfect way to put it. Normal healing gets replaced by this persistent damaging process.
That makes the aberrant part crystal clear. Yeah. So if it's a dysfunctional neighborhood, who are the specific troublemakers? Which immune cells are driving this bad behavior and leading to the scarring?
Right. So the researchers use some really advanced techniques, things like spatial transcriptomics and imaging.
Fancy mapping tool.
Yeah, basically cutting-edge ways to see which genes are active and how cells are interacting right there in the actual lung tissue. And they pinpointed a central role for two specific types of immune cells. And importantly, these are lung residents.
Meaning they live there permanently, not just visiting.
Exactly. They've set up shop in the lung tissue. And these are CD8 plus T cells and macrophages.
Okay. CD8 plus T cells, the killer T cells, right? Usually key for fighting viruses. And macrophages, the big cleanup crew of the immune system.
That's them.
They sound like they should be the good guys here, helping clean up after the virus. So how on earth are they causing trouble?
Well, what's really fascinating and kind of disturbing is the specific interaction and, critically, the timing. The CD8 plus T cells, which normally mount this powerful but temporary attack to clear a virus.
Right, they do their job and stand down.
Exactly. But here, they start persistently releasing two key inflammatory signals. Molecules called IFNO, that's interferon gamma, and TNF, tumor necrosis factor.
Persistently,
Yeah.
But they don't shut off.
They don't shut off. Instead of being a short-lived signal for the acute phase, these molecules act like constant incorrect instructions within the lung. They keep signaling danger long after the initial threat is gone.
Like a faulty smoke alarm that just keeps shrieking even after the smoke is cleared, basically stopping anyone from getting back to normal.
That's a great analogy. Exactly. And then these persistent signals from the CD8 plus T cells, they stimulate the local macrophages.
It's a cleanup curve.
Right. They stimulate them to chronically release another powerful inflammatory signal, a cytokine called IL-1, interleukin-1 beta.
Okay, another signal. And chronically is the key word again.
Absolutely critical. It's not that helpful temporary burst of inflammation you need to fight infection. It's this sustained, low-level, continuous signal that persists long after the acute infection is resolved. And this constant stream of IL-1, that seems to be the central driver of the ongoing lung damage.
Okay, well, let's just pause for a second and recap this, because it feels like we're getting to the heart of it. This is dense, but really important. We've got these lingering lung problems after viruses, especially PAC.
Right.
And the core finding is this aberrant immune-epithelial progenitor niche, this messed up healing environment in the lungs.
That's it. And inside that abnormal neighborhood, specific immune cells, these permanent residents, the CD8 plus T cells and macrophages, they get caught in this damaging feedback loop.
Right. The T cells keep shouting danger with IF-now and TNF, which makes the macrophages continuously pump out this other inflammatory signal, IL-1.
Exactly. This constant, unhelpful inflammatory crosstalk is really at the core of the problem.
So, okay, this constant alarm bell, this IL-1 blaring away, what does that actually do to the lung's own ability to heal properly? Because it sounds like it's not just inflammation anymore, it's actually messing up the repair job itself.
You've absolutely nailed the crucial consequence there. This chronic release of IL-1 is so impactful because it directly impairs the lung's ability to regenerate its most delicate structures, the air sacs, the alveoli.
Or the oxygen exchange happens.
Precisely. Now, normally after an injury, there are specialized lung cells called AT2 cells, alveolar type 2 cells. They mainly make surfactant, which keeps the air sacs open, but they also act as key progenitor cells.
The repair crew we talked about.
They're the foundational stem cells for that area, yes.
Hmm.
Their job is to divide and then mature into AT1 cells. And AT1 cells are the really thin, flat cells that form the huge surface area needed for efficient gas exchange. Think of AT2s as the bricks and mortar.
And AT1s are the finished functional wall.
Perfect. Yes.
Yeah. So AT2s are the master repair cells, meant to rebuild the parts that let us actually breathe properly.
Exactly. And in healthy healing, AT2 cells go through specific intermediate stages to become those AT1s. One critical intermediate state involves cells called DTPs, damage-associated transient progenitors.
Okay, DTPs.
Now, interestingly, some inflammation, like an initial burst of IL-1, is actually supposed to help kickstart this process. It gives the AT2 cells the go signal to begin repairing.
All right, a little nudge to get started.
But, and this is the crucial finding highlighted here, chronic inflammation driven by that persistent IL-1. It actually prevents the final essential maturation step into healthy AT1 cells.
So it starts the process, but then blocks the finish line.
Precisely. It's like the signal to start rebuilding never turns off, but the signal to actually finish the job properly, to build a functional wall, is somehow blocked or never given.
Wow. So the initial push is there, which seems good, but the chronic signal craps the cells. It gets stuck in this loop and stops the healthy repair, leaving the lung unfinished, faulty.
That's exactly it. It leads to what the study calls an aberrant accumulation of DTPs. These precursor cells get stuck in this abnormal transitional state. They just can't fully mature into healthy working AT1 cells. And this abnormal growth, this perpetually stalled development, is what they term dysplastic epithelial progenitors. They're trying to repair, but they're doing it all wrong. They're creating faulty building blocks, an incomplete structure, not functional lung tissue.
And that's what drives the fibrosis, the scarring and stiffness that makes breathing so hard for people long after the virus is gone.
Bingo.
It's a continuous cycle of botched repair. It's a fundamental structural issue, not just some leftover inflammation.
That's absolutely right. That constant abnormal signaling from the immune cells, the CD8 T cells and macrophages stuck in their loop, creates this environment where the lung's own repair crew becomes dysfunctional. Instead of smooth, effective regeneration, you get this persistent scarring and consequently severely impaired pulmonary function. It's a fundamental breakdown in the body's healing playbook.
Okay, so if we understand this mechanism, now we know who the rogue players are, the signals they're sending, and how that messes up the lungs repair crew. Can we stop it? Can we interrupt this cycle? What does this research suggest for new therapies? For actually helping people who are struggling with these awful long-term symptoms?
This, I think, is the most hopeful and potentially impactful part of the whole study. They demonstrated in their models that therapeutically blocking either the IFNO and TNF,
The signals from the T cell.
right, or blocking the IL-1,
The signal from the macrophages.
that doing either of those things markedly improved alveolar regeneration. And crucially, it restored pulmonary function.
Wow. So they found a way to basically cut the wires, to turn off that faulty alarm system or block those bad instructions getting to the repair crew.
Effectively, yes. They showed you could intervene in that faulty communication loop.
But here's the really critical part, isn't it? And this feels so important for the millions affected by PSAO. This isn't about treating the initial infection when you're acutely ill, right? This is about intervening later.
Correct. And that is a massive distinction, offering immense hope. Unlike so many treatments that have to be given really early, during the acute phase, maybe the first few days or weeks,
Yeah.
this research specifically points to strategies to rescue this fibrotic disease after the acute illness has resolved.
After the virus is gone.
Long after, potentially. This is a huge breakthrough because it directly tackles a major unmet need in managing PSC and maybe other post-viral conditions too. It offers real, tangible hope for people already suffering from these long-term consequences. It suggests that maybe, just maybe, lung function could be restored even months or possibly years down the line.
That's incredible. So what does all this mean for you listening out there, for the future of treating these really stubborn post-viral conditions that have just impacted so many lives across the globe?
Well, I think it means that by really meticulously understanding the precise cellular conversations, the chemical signals driving chronic disease, we can actually develop targeted, effective interventions.
Not just throwing things at the wall.
Exactly. This study specifically fingers these key inflammatory pathways involving those lung-resident CD8 plus T cells, the macrophages, and that downstream IL-1 as central culprits in lung scarring after viruses. And critically, it shows that blocking these specific pathways can actually promote healing and regeneration, even long after the initial insult. That gives us a clear, exciting path for developing future therapies.
It's just mind-blowing, really, to think that the long-term health outcome after a virus could hinge on these tiny, specific interactions between immune cells and the lung's repair cells.
It is.
Imagine if just understanding these microscopic conversations, these specific signals going wrong, could unlock better, maybe even complete recovery for millions. It really makes you wonder, doesn't it? What other unmet needs are out there in other post-viral syndromes or even other chronic diseases we struggle with? Are we missing similar things because we haven't looked closely enough at this persistent cellular chatter?
That's a really important question this research brings up for all of us. How could a deeper dive into these intricate cellular feedback loops, where one cell's signal chronically affects another's function, creating these self-perpetuating cycles, how could that revolutionize how we approach chronic illness way beyond just viruses? Could these kinds of aberrant niches, these dysfunctional microenvironments, be hiding in plain sight in other conditions we find hard to treat, just waiting for us to figure out their secrets?
So much to think about there. What insights from today's deep dive really resonated most with you? And what questions does this research spark about long-term health after maybe even common infections? We genuinely love to hear your thoughts. Until next time, keep diving deep.